All right, you guys, we're at unit 3.2, still structure and function of DNA, but we are moving to part 4, which is on protein synthesis. All right, so there's quite a bit to get through in this section. We have to talk a little bit about another type of nucleic acid called RNA before we can even talk about making protein. But you guys are smart kids, I believe in you, so we are gonna cover all of this in one video lesson, all right? Great. So first thing, what is RNA? So RNA is, like I said, it's another nucleic acid. It stands for ribonucleic acid. So it's a nucleic acid that includes the sugar ribose. It is single-stranded, as opposed to DNA, which remember, DNA was a double-stranded nucleic acid, and it had deoxyribose. Now, RNA is single-stranded, and it contains ribose as its 5-carbon or pentose sugar. Still has a phosphate, but the bases are a little bit different. Remember, in DNA, with a nucleotide, you had deoxyribose, phosphate, and nitrogenous base, and the bases include adenine, guanine, cytosine, and thymine. Well, in RNA, it could be a little tiny bit different. In nucleotide, instead of deoxyribose, we have ribose, we have our phosphate, and we have adenine, guanine, and cytosine, but there is a change. There is no thymine in RNA. Instead, we have URACIL. URACIL is the base that replaces thymine. It does all the same things that thymine does. It is a pyrimidine. It can form 2 hydrogen bonds, and it is complementary to adenine. But we see uracil in RNA, we do not see thymine. Now, there are 3 types of RNA that we are going to mention for protein synthesis. We have messenger RNA, which is more linear. We have ribosomal RNA, which is part of the structural makeup of ribosomes. Remember, ribosomes are a tiny organelle that helps to make protein. And then there is tRNA, or transfer RNA, which kind of makes this weird loopy shape. And what it does is it brings amino acids so we can use them to build a protein. And we're going to talk a little bit about each one, starting with messenger RNA, or known as mRNA. What this is it is a copy of the information from the DNA. Now, it's not the whole DNA copied. It's a section. Specifically, it's a gene. So we copy a single gene in the form of messenger RNA. Now, the reason why we do this is because DNA is located inside of the nucleus. Well, all the stuff to make protein is located outside the nucleus in the cytoplasm. So the point of mRNA is to get just the pertinent information, so just the information for this one gene that we want to create the protein for. So we're going to take just that information, we're going to move it outside of the nucleus to the cytoplasm, to where the resources for protein synthesis are. All right. Then we have ribosomal RNA, or rRNA. And like we said, this is simply a structural component of ribosomes. Ribosomes are made up of rRNA as well as proteins. So the protein and the rRNA come together to form this tiny organelle, and this organelle is going to have a huge function, a huge role to play in protein synthesis. Right, Ricky? Yeah. Sorry, I have my dog sitting next to me. And then here we have transfer RNA, or tRNA. Now, the job of tRNA is to read the code in the mRNA and bring the appropriate amino acid in order to build the amino acid chain. Now, we'll talk a little bit about how it does this using bases within the tRNA and the mRNA. But just to give you a kind of overall view, this would be the transfer RNA, and notice at the top end, it is carrying an amino acid with it. So here we have a general view of everything going on in protein synthesis. We have the mRNA down here. We have the ribosome, which includes the rRNA, and then these purple-blue squiggles, those are the tRNA, carrying the amino acids down so they can be connected into a chain, and that will be used to make our protein. All right, there's that. Let's talk a little bit about the central dogma. Now, a dogma is a core belief. So a central dogma is the core belief of essentially molecular biology. So this is the foundation that everything in molecular biology stems out of. You guys ready for this? All right, central dogma of molecular biology. DNA is used to make RNA, which is then used to make proteins. Now the reason why this, it seems so simple of a statement to be the foundation for an entire branch of biology, but a lot of molecular biology looks at proteins and how they do their job. Well, proteins are some of the most versatile molecules in our body. They express all of our traits. They build our tissues. They defend us against bacteria and viruses. They catalyze reactions. They are enzymes. Come on, there's all these different uses for proteins. Well, then the question becomes, all right, if they're so essential for us to be alive, how do we get them? Well, we get them from the DNA and the RNA. Remember, the DNA is the entire code, the entire blueprint to make you. What we do is we take that entire code and we copy a section of it, a gene. And that's what we use to make the RNA. And then we take the information from the RNA, bring it over to the cytoplasm where the ribosome and the tRNAs are located, and we can now use those resources to create a protein. So this is just kind of very, very, very simplistic drawing. We begin with the DNA. We transcribe it to form the messenger RNA. It gets translated to form an amino acid chain, which will then be used to form a protein, which will express a trait or do some kind of important job in your body. All right, let's talk a little bit about vocab before we get on with the steps of protein synthesis. There are two terms that you're going to hear me use a lot in this video lesson, as well as in our assignments and everything like that. First one is codon. A codon is a group of 1, 2, 3 bases in the mRNA. So there are 3 sequential bases. For instance, here, GA, and then this box, whether it's GAA or GAG. We have GGU, ACA, UUG. All of those, actually I'm reading it backwards, but all of these are codons. So there are 3 sequential bases in the mRNA. And these 3 sequential bases will code for a few things. They'll either tell us which amino acid should go next in the protein, or whether or not to start building the amino acid chain, or whether to stop building the amino acid chain. So it's either going to be a start codon, a stop codon, or code for an amino acid. Now remember, it's the tRNA that brings the amino acid and puts it in its correct place. Well, how does it know where its correct place is? Because on the tRNA, there's something called ANTICODONS. And ANTICODONS are complementary to codons. So they are a group of 3 sequential bases in the tRNA that are complementary to the codons. And since they're complementary, that means that they will line up with the codons, and each tRNA is unique in which amino acid it's going to bring. So the codon determines which anticodon can line up with it, and on the other end of the tRNA, that's where the amino acid is. If you look here, ACA is our codon. This tRNA knows to line up with it because the bases in its anticodon are complementary. What's complementary to A? U. Remember, URACIL is in RNA. What's complementary to C? G. What's complementary to A? U. So here we have our anticodon. UGU. So that's how it knows where to line up. And on the other end of it, it has a very particular amino acid. Now that amino acid gets added in the chain. Another tRNA can come in, complementary. A is complementary to U, C with G, C with G. And that's going to bring this next amino acid and put it in this correct spot. So it's all based on lining up those complementary codons and anticodons in order to get the amino acid in the right position. All right, so first, let's talk about transcription. Now, this is the process of making the mRNA from the DNA. So remember, DNA is the entire blueprint. We don't want to copy the entire blueprint. We just want to copy this one gene. And that's what transcription does. Now, the process of transcription is almost identical to that of DNA replication. We have helicase. Helicase is going to unwind the DNA and separate the two strands. We have RNA polymerase, which is going to read the template strand of DNA and start attaching and building the complementary bases. But this time, since it's RNA polymerase, it's going to be making RNA nucleotides as opposed to DNA. So that means the nucleotides are going to have ribose and possibility of uracil rather than thymine. So the RNA polymerase matches up the RNA bases to the DNA template. Again, going off the Chargaff's rule, putting the complementary bases together until a stop codon is reached. Once the stop codon is reached, then the DNA is going to separate from the new RNA. The DNA is going to reconnect with itself, so it's going to reform those hydrogen bonds and then retwist back up into its helical shape. That's kind of what we see here. We have the blue on the top and bottom. That is the DNA. It gets unwound and opened up, and then we start building it, this RNA strand using RNA polymerase. And it's just going to read along the DNA, making complementary bases. You notice U to A, CG, AT. Because yes, thymine is in DNA. That's why we see thymine down here. But we don't see thymine up here because there's no thymine in RNA. Any place you would expect to see a thymine is replaced with U for uracil. So A to T, C, G, GC, CG, AT, GC, UA, CG, AT, UA, GC, UA, GC. When we reach the stop codon, everything disconnects, the DNA curls back up, and we are left with our mRNA. Now, the mRNA is going to leave the nucleus through one of those nuclear pores in its membrane, and it's going to go out into the cytoplasm. Once it's in the cytoplasm, it gets picked up by the ribosome. So you might have noticed in some of those pictures, it kind of looked like two parts. What happens is you have a small subunit and a large subunit. So the small subunit is going to attach first, and then the large subunit will attach later. This small organelle is the one that's responsible for helping to make proteins. Now what happens is, well, proteins are another macromolecule, and so they're made up of repeating smaller subunits called monomers, and for proteins, those monomers are called amino acids. Now the ribosome has two slots in it that the transfer or tRNA can slide into in order to help it line up with the proper codons on the mRNA. So the ribosome holds onto the mRNA and then transfer the tRNAs with the matching complementary anticodons will line up. They'll slide into those slots on the ribosome and they will be able to line up with their complementary codons. On this end, they have an amino acid. Now that amino acid is going to be added to the chain of amino acids, and it's going to keep going and going and going. And it's going to keep going and going and going until you, once again, you reach the stop codon, and then everything disconnects. So the anticodons line up with the codon on the mRNA according to those base pair rules, the Char-Gas rules. Once it attaches the amino acid and the next tRNA drops in, it will disconnect and it will actually leave the complex. It will leave the ribosome and it will go back onto the cytoplasm to get another amino acid. So here we have the mRNA, the ribosome is connected to it. Notice how here we have UGG, and complementary to that was ACC. Now do you see an amino acid at the top? No, it was actually originally connected to this one. It brought this amino acid down. But right now, once these both were in the ribosome, this guy disconnected and left. Right now, this tRNA is holding onto the amino acid chain. See, there's his amino acid. And you know that he's supposed to be there because UUU is complementary to AAA. The second tRNA drop down, CUA, is the anticodon, GAU is the codon, and there is its amino acid. Once this bond has formed between these two amino acids, securing this guy to the chain, then this bond here is going to break between the amino acid and this tRNA. This tRNA is going to leave just like this guy did. The ribosome shifts over, and this one is going to now drop down next to this guy. AAG, UUC, and that will attach up. His amino acid will attach with that one, causing this bond to break and that one to leave. All right, step 3, protein synthesis. We're not going to get into a whole lot of detail on protein synthesis. There's a lot more we can discuss about it, but we're just going to very, very briefly summarize the basics. So protein synthesis is the process of translation. It's the process that occurs after translation. Now, remember we said translation will occur until a stop codon is reached. Once the stop codon has been reached, the ribosome lets go of the mRNA, the tRNAs let go, and you are left with an amino acid chain. Now, this is also called a polypeptide chain, which is why we also call the bonds between the amino acids peptide bonds. So the polypeptide chain at that point is non-functional. Think of it like a long piece of yarn. I mean, it's fun to play with, but you know, nothing can be done with it. However, if you take that long piece of yarn and knit it, you can turn it into a hat. A hat has a function. It keeps your head warm in winter. That's kind of what is going on in step 3 of protein synthesis. This is protein folding. Sorry, I should say protein folding. Anyways, protein folding. So we take that polypeptide chain, that amino acid chain, and we have to either fold it into its correct 3D shape or bring it to the rough ER, the rough endoplasmic reticulum, because remember, the primary function of the rough ER is protein modification. So it can modify the protein before it can be folded up. Now, just like how a piece of yarn or a long thing of yarn has to be knitted into a hat for it to have a function, the same way with the protein. The chain of amino acids, the polypeptide chain, does not have a function until it folds up into a 3D shape. Once it's folded up, first off, the actual order of the amino acids and the placement of the different amino acids will determine what shape it makes once it's folded up. And the shape that it makes when it's folded up will determine its function. So that's why folding it is super duper duper important. So this is just kind of showing you the basics of it. We're not going to get into this much detail in class, but here is the amino acid chain. Call that the primary structure. It can either form these alpha helixes or these beta pleated sheets. And then it'll form this tertiary structure. And then it will combine with others to form a larger protein, like here. We took these, used it to make a subunit of hemoglobin, add 3 more, so you have a total of 4 that are connected. You now have hemoglobin, and that is a functional protein. Let's see. So overall protein synthesis picture, use the DNA and use that to create mRNA. We call that process transcription. The mRNA leaves the nucleus, goes onto the cytoplasm where it's picked up by a ribosome, which helps the codons in the mRNA and the anticodons in the tRNA to line up in the proper way. This allows the amino acid that's on the other end of tRNA to get close enough to form these amino acid chains. Once they're done, the protein leaves and it is folded up into its correct shape. Now you've heard me talk a lot about, well, the amino acids. How do we know what amino acid goes where? How do we know the order? Well, by using this chart here, you can actually figure out the amino acid sequence. If I give you mRNA, you can use this chart to figure out the order of the amino acids. Now, how you read this, first of all, always use the mRNA, the messenger RNA with this chart. It's called an mRNA codon chart for a reason. Do not use the tRNA with this chart. You will get the wrong answers. Now remember, mRNA, we have those codons, and the codons are sequences of 3 bases. Well, what you do is you start in the middle with the first base. So let's say it is, a codon is GAU. We have GAU. Start in the middle, there's G, then go to the next ring, A, and then U, aspartic acid. So the mRNA codon of GAU codes for aspartic acid. If it is CCC, proline. If it is CAA, glutamine. CAC, histidine. ACG, threonine. UGG, tryptophan. Now, there's a couple specific ones I also want to point out. UGA is one of those stop codons. We have 3 total stop codons. We mentioned UGA. We also have UAA would be stop, and UAG would be stop. Remember, I also said there's a start codon. That is methionine. Methionine is a special amino acid that creates a reaction that starts attracting all the other tRNAs to that spot. So the start codon for any gene should be AUG. This is going to attach a methionine, which is an initiation or an initiator molecule, meaning that it then signals that we have to start translating this mRNA, and it's going to bring all the other components down to that spot. So methionine is our start codon. All right, let's talk a little bit about additional resources. I would highly recommend that you check out my protein synthesis video. I'm not just saying that because I made it, but as you can see, I actually made this big paper structure that shows how protein synthesis occurs. So I walk through all the different steps from DNA synthesis, from transcription all the way to, using the charts. And then other videos, we have the amoeba sisters, DNA versus RNA, so comparing the two nucleic acids. We have amoeba sisters, protein synthesis. We have amoeba sisters, how to read the codon chart. So in case you're curious and you want to make sure. Amoeba sisters, protein structure and folding. Yes, I love the amoeba sisters. I think they do a great job of explaining everything, breaking down the different parts of each process and structure, and also kind of making it visually appealing as well. McGraw-Hill animation, DNA transcription and translation. And then we have Don't Memorize, Central Dogma of Life. So definitely check out some of those to help further your understanding of this process. In the meantime, you guys just stay awesome, stay amazing, stay safe, stay curious, and I'll see you later. All right, you guys, I also will post the link in the video description so you can access the additional resources. All right, you guys, take care of yourselves.